The next speaker is Alex Vallmitjana. Alex, I don't know if you are connected. Think so. Maybe you can turn on your camera and we will ping you. Can you hear me? Dr. Alexander Vallmitjana is a colleague and an old friend. We share desk at the UCI when we were doing both our postdoc. Alex Vallmitjana is a physicist in training, and then he dedicated his PhD and in bioimaging analysis. And after a postdoc at the University of California, he moved to biomedical engineer and data scientist in a startup, Arvetas bioscience startup with base in California, Irvine. And I wanted to bring his experience in moving from academia to a challenge and very, intricate a path, which is going to be your own boss. So I would like to give you, basically, I will pass the word to you.
Thank you. Thank you for the introduction. Let me share the screen. All good?
No, we are watching at your desk. Not your desk, the presentation, not the presentation mode, basically the, oh, now yes.
Okay, yeah, once again, thanks for the introduction. Yeah, I'm kind of the representative of the startup world, and I title my talk, "Enjoyment Will Take You a Long Way," because I think that is what, when they asked me, what is the key to your path, I think that was what drove me all along. It's like I'm more driven by present time. So as long as I'm having fun, I can continue doing what I'm doing forever. And I think without even choosing, I kind of found a spot in my career. I know that this is not applicable to anyone. Some people are more goal-oriented, but at least that's what worked for me. As Lena stated, I started at the University of Barcelona as a physicist. Curiously enough, just like Michelle, I am also a third-generation PhD, and my dad was also a physicist. So he's probably the first inspiration and love for science. I was always going to go into astronomy. I had a passion for astronomy, and with like a couple of friends, we used to go out into the mountains and use this 30-year-old equipment from our parent generation. This was already in the digital era, but we used to, take this old equipment with really good optics, but cumbersome film. You're having to develop the films after each. You never got to see what you were imaging, but nevertheless, we got pretty cool images. And this is what started interesting me into image processing. At that time, we would digitalize these images, and then a posteriori, we would do Photoshop to enhance the colors, balance out the levels. And I soon realized that in undergrad that this could be done automatically, this could be done programmatically. So that's what got me interested for the first time in image processing in the scientific world. I was offered a possibility to start a PhD in image processing. It just so happened to be that it was applied to biomedical engineering. So I got a PhD in biomedical engineering for the Universitat Politècnica de Catalunya. At that time, I just saw it as, to me, it's the same stuff. It's basically solving problems in imaging, doing, finding exciting solutions to stuff that people aren't, they're in a pickle, and say, for example, the sciences is irrelevant today, but they would say, oh, I have to detect all these structures in cells and, apply some algorithm to detect all the objects and find the distribution, quantify how they're distributed in the images. Typical examples of image processing, cell segmentation. You have a culture of cells, and the scientist is interested in figuring out where's the border between each cell, so developing algorithms for cell segmentation. Detecting objects, classifying them into types of objects, colocalization of detected objects, that they appear together, that they not. Well, this was a super exciting project in which we did motion tracking applied to calcium waves, and this is a heart cell. This is an unhealthy heart cell. A heart cell like this would trigger fibrillation if you have enough of them together in the heart. And this also led to, as a continuation of the video tracking work, to something completely different, but super fun as well, which was this very important academy, tennis academy that needed video tracking for teaching the students. And yeah, basically developed this software, which is not only video tracking, it also does projection into real life, like real world coordinates, which is very useful. Soon after that, this is in 2017, I got the possibility of moving to Irvine, to the Laboratory of Fluorescence Dynamics under Enrico Graton. And that is where I met Lionel. And that is where I, for the first time, got some experience in hands-on equipment. For the first time, I was actually building the microscopes, not only analyzing the data that came from the microscope. So this is the instrument that I helped build and I maintained for 3 years at the LFT, very versatile, custom-built microscope. And I continued working in image processing, in this case, developing more tools. This is the phaser approach, is one of the methods best known from this lab. So I've continued working on some of the image processing techniques. Again, cell segmentation, in this case, using clustering methods from machine learning. And one of the most important projects that I worked with was together with the pharmacology department, where we developed a technique to label many species of RNA transcripts and image them using several imaging techniques in a single shot to detect the spatial location of each individual one of them. And although it's basic science, it has direct applications into diagnostics. And so we got a patent out, and through that, this is where the startup became. The idea being to develop a imaging-based diagnostic tool, which would be a very early diagnostic for cancer, when cancer is not even there, only the first markers appear and you already can detect them. So we co-founded this company in the summer of 2020, actually moved to the research park, to the startup incubator building in the summer of 21. And that's where I've been till now. It's a very cool environment because it's a building where, they have like dozens of startups together. You have your own office, then you have common spaces. So you get to meet with a bunch of people who are working in very different environments, but at the same time, it's the same startup world. In our case, as I said, we're doing, it's the field of spatial omics. So detecting expression of protein and RNA in space, in a biopsy. So in the tissue, you find out where they are and what's their relationship in space. And maybe I have brought some details on the startup world, what to expect. So first, it's a very high risk environment. Startup success rate is below the 10%. It's even worse in tech companies. When I started, the estimate was 5% success rate for us. The failure reasons, typically market demand. So you think you have a cool idea you want to develop, but you haven't done enough market research. So it's either not needed or people are not prepared to pay whatever you're asking for it. The other typical reason is you run out of money. At some point, you need money. So if you don't develop fast enough and you run out of money, you're too slow, you're done. And a very important thing is also team culture. You need a core team with which you're happy, you're enjoying yourselves again, and you can trust each other, right? How do you get your money? Well, typically it's private funding, the seed financing. It's either some wealthy individual that's through some company or even someone close known person. In this case, our scientific co-founder had a friend back home in China who's a wealthy individual who gave us the initial amount that allowed us to start the company. And then you need successive rounds. Typically, people go through venture capital firms, which is a company that deals with private money as well, and have these high risk investments. They will give millions to a startup company. Obviously, at the getting a fraction of the company, so getting shares, they will own part of your company. And what we have done in our particular case is the Sbir program, which is a national public program for small business innovation research, I think it is. in which it's basically you apply for a grant to the government, just like in academia, which makes our work in the startup very much like academia. I mean, I've spent quite a bit of time of writing grants, and in this case, the Sbir program works with milestones. It's not like they give you the lump sum money and then you develop your work. It's a bit the opposite. You say you're going to do some milestones, and then after each successive quarter, if you have completed your milestones, they give you a fraction of the money that you were awarded. We're in fact in the final quarter this month, so I'm right now writing the report for them, for the NIH. This is in the National Cancer Institutes, NCI. Possible outcomes of a startup. Typically within less than 10 years, you're either, well, yeah, typically you're either done for in the first 1 or 2 years. If you succeed, you get acquired by a bigger company, and the most rare case is to go public. So you become a big enough company that offers shares to the general public. One of the bad things, the salary is low compared to going into industry. Yeah, the median salary across all startups in the US is that of a postdoc. As a compensation, you do get shares in the company, especially if you're a co-founder and you own the company, right? So that's kind of the reason why it's a high risk but high reward. If good things happen, you may make a lot of money down the line. So yeah, to finalize what to expect, as I said, high risk, high reward. Also, like Lionel said, being your own boss, this is a good thing. Like in academia, it's a bit like that as well. Very steep learning curve, because it's, as well, the last point is you're wearing many hats. You have to do all kinds of things. You can be, purchasing, as I said, writing grants. You can be recruiting new employees, so doing interviews. The whole company is 3 people in our case. It's a unique and ever-changing path. You're always doing new stuff, and you're always submitted to very different environments, which is, I mean, can be fun, and for sure it's a very enriching experience, because it exposes you out of your comfort zone. So yeah, that's about it for me. I'd like to thank some of my mentors in my career. My dad, for starters, my optics professors and image processing and coding teachers, and my PhD advisors, and Rico Raton, my postdoc advisor, with whom I did the work that led to the company, and our current CEO and leader of our team, Alan. Thank you very much.
Thank you, Alex.